Hi students, we have completed the lesson applications of biotechnology both for pure science and also for bio students. Now we have to pass on to the next one, that is chapter 2, human reproduction. Now what is the importance of human reproduction? The human reproductive system is responsible for or essential for the survival of the species. The survival of the species depends on the human reproductive system. Now what are the four major functions performed by the human reproductive system? Number one, production of gametes, the units of reproduction. Male and female gametes, namely the sperm and egg. That is number one. Number two, transporting the gametes, particularly the male gamete, to the female reproductive tract. And we have to sustain these gametes, maintain the gametes without being damaged. That is called transport and sustain gametes, particularly the male gametes from the male individual to the female reproductive tract. Number three, nurture the developing offspring. Just actually looking after. Nurturing means looking after. Nurture the developing offspring. That is in the womb of the mother. The next one, what we call the produce hormones. Now the male and female sex glands, namely the primary sex organs, ovaries and testes, they produce different hormones. For example, the male gonad, the testis produces androgen. The female gonad, namely the ovary, produces estrogen and also other hormones during pregnancy. That's about what are the four major functions of the reproductive system. Now let's pass on to what are the various steps involved, the major steps involved in reproduction. We have different steps are involved in reproduction. We'll see one by one. Now what are the major events of reproduction, that is reproductive events. The first step during reproduction is gametogenesis. The word genesis refers to the formation, development, etc. Genesis, the word meaning formation. It is nothing but the formation of gametes, namely the male gametes, the sperm, and the female gametes, namely the ova. So both are being produced by means of two events. The formation of sperm is called spermatogenesis. Formation of sperm is called spermatogenesis, and the formation of ova or egg is called oogenesis. So we have gametogenesis, two types. One in the case of male called spermatogenesis, another one what is called oogenesis. Spermatogenesis, the formation of male gametes or spermatozoa. Oogenesis, the formation of the female gamete or ova or eggs. So that is one. After the formation, during copulation, the sperm from the male body is transported into the female reproductive tract. That process is called insemination, transfer of the male gamete, namely the sperm from the male individual into the female reproductive tract. That is called insemination. The next one, in the female reproductive tract, we have the fusion or union of male and female gametes to form a single cell, what is called zygote. The fusion of female and male gametes to form a single cell called zygote is called fertilization. It occurs in the female reproductive tract. Now the female gamete, what is called the egg, is in nature, and also the male gamete N. Now the resultant cell, the single cell, is diploid. So the union of male and female haploid cells to form a zygote, which is diploid, and that process is called fertilization. Now once actually the single cell, what is called the zygote, is formed, the zygote undergoes repeated mitotic divisions. Now this is the zygote, which undergoes repeated mitotic division to form a multicellular structure, and that structure is called blastocyst. So this is a multicellular structure, what is called blastocyst. In the case of other animals, now the formation of actually blastula, a spherical ball of cells by the repeated division of zygote, that occurs. But in the case of human beings, now the blastula is called blastocyst. It's not called blastula, blastocyst. So cleavage is nothing but the repeated mitotic division of the fertilized egg, namely the zygote, into a multicellular structure, what's called blastocyst. It is also called segmentation process. It is also called segmentation. So we have the cleavage occurs by means of mitosis. Conversion of single cellular diploid zygote into a multicellular diploid, what we have actually the blastocyst. The cells of the blastocyst are called the blastomeres, which are also diploid in nature. Now the next one, implantation. There is also a technical word for implantation, what is called nidation. It is also called by name nidation. This is nothing but the attachment of the fertilized, what is called the blastocyst, or we can say zygote. Simply we can say the attachment of the blastocyst to the inner wall of the uterus. That process is called implantation. Attachment of blastocyst to the inner wall of the uterus, what is called endometrium. The inner wall of the uterus is called endometrium. We'll see under the female reproductive system. That process is called actually implantation, otherwise called as a nidation process. Then formation of placenta. Now it is a vascular structure that connects the developing fetus with the uterine wall of the mother for exchange of nutrients between the mother and the fetus. So placenta is a vascular structure that normally connects the fetus and the uterine wall of the mother. The main function is exchange of nutrients and gases. Okay, that's about the placenta, formation of placenta. Once formed, it is also acting as an endocrine gland. Then gastrulation. If you are taking the blastocyst, all the cells are similar, diploid, no differentiation. But during gastrulation, what is happening? The blastocyst gets converted into three-layered gastrula. Three-layered gastrula. And this process is called gastrulation. This process is called gastrulation. So conversion of, we can say, single-layered blastocyst. The meaning for single-layered blastocyst, though it is formed of many layers, is being formed of only one type of cells. Now during gastrulation, we have received what is called gastrula. Here it is three-layered. The meaning for that one, it contains three germinal layers: ectoderm, endoderm and mesoderm. That's why we are using the word three-layered gastrula. It's not representing the number of layers, but the different layers: ectoderm, endoderm and mesoderm. Then organogenesis. It is nothing but the process of formation of various tissues, organs and organ systems from the three germinal layers, either from the ectoderm or from the endoderm or from the mesoderm. For example, the ectoderm gives rise to various nervous structures and related sense organs. The mesoderm gives rise to various parts of the body, just like muscles, the bones, the circulatory system, etc. Then we have the endoderm gives rise to some endocrine glands, for example thyroid, and also the digestive tract. So that's called organogenesis, formation of various tissues, organs and organ system. That's what is called as organogenesis. I mentioned already, genesis means the formation. Then parturition. Now once the embryo is completely developed, it is being expelled from the womb of the mother. That process of expulsion of the fetus from the womb of the mother is called what is known as parturition. It is nothing but childbirth. It is nothing but childbirth. So these are all the various steps. Normally we have sub-steps also, but the major steps involve the events of reproduction or gametogenesis, then insemination, transfer of sperms from male to female body, fertilization, union of male and female gametes to form a diploid zygote, cleavage, the segmentation of the fertilized egg into a multicellular structure, what is called blastocyst, implantation, attachment of the blastocyst to the inner wall of the uterus, then formation of placenta, the vascular connection between the uterine wall and the developing fetus, gastrulation, conversion of blastocyst into what is called gastrula with the three germ layers: ectoderm, endoderm and mesoderm. Then organogenesis, formation of tissues, organs and organ systems, and parturition, nothing but the childbirth. Now how are these functions carried out? We have to see. Now the functions of reproductive system are carried out by number one, primary reproductive organs. Number 2, accessory reproductive organs. Now the primary reproductive organs include mainly the ovary and testes. Ovary is responsible for the production of ova or eggs by oogenesis. Testis is responsible for the production of sperms, the male gamete, by spermatogenesis. That's the role of ovary and testis respectively. Now we have certain hormones secreted by the pituitary gland and also hormones secreted either by the male reproductive system or the female reproductive system are responsible for certain functions. Now what are the functions of hormones of pituitary glands and gonads? Number 1, development of secondary sexual characters. We have primary sexual characters, namely the presence of testes and ovaries, by which one cannot identify whether the individual is male or female, because in human beings we have sexual dimorphism is exhibited. So by means of external genital organs only one can identify whether the individual is male or female, and such characters which are used to identify the male and female externally morphologically are called secondary sexual characters. For example, beard in the case of male and mammary glands in the case of female. Just I'm giving only one example. So the hormones are responsible for the development of secondary sexual characters. Number 2, responsible for the maturation of reproductive system. Number 3, responsible for regulating the functions of reproductive system. 1, development of secondary sexual characters. Number 2, the maturation of the reproductive system. Number 3, the regulation of functions of what we have the reproductive system. These are all concerned with the primary reproductive organs. What are the functions of accessory reproductive system? They're mainly concerned with, you know, that one to transport and sustain gametes. This is one of the functions I mentioned already under the reproductive functions. So the accessory reproductive tracts, they are responsible for the transport and to sustain the gametes. From the male to female, they are being transported. So transport and to sustain gametes. And also for nurturing the developing embryo, for nurturing the developing offspring, or nurturing the developing fetus. These are the 2 functions: nurturing, looking after, till birth. So the accessory reproductive organs, for example, the uterus is an example for accessory reproductive organs for bearing the fetus that is undergoing development. So one, the transport and the sustain the gametes, maintaining the gametes without being damaged. The second one, nurturing the offspring, the one which is developing in the womb of the mother. So that's about what we have the functions of reproductive system. They are being carried out by what we have the primary reproductive organs and accessory reproductive organs. Their role, what are the roles played by these 2? Okay, now we have to go for the next one, the human male reproductive system. Now let us discuss the human male reproductive system. If you are taking the male reproductive system, it is formed of the primary, that is what we have, sex glands or primary sex organs, a pair of testes. Number 2, the conveying structures, accessory ducts. Number 3, the accessory glands for the production of some fluid. Then external genitalia, the sex organs present outside for identification of male. Now let's take the first one, testes. And now you know that one, in the case of all mammals, exception is there, we have the testes are placed inside what we have are located or lie inside a sac of skin, what is known as a scrotum. So the testes are ovoid bodies lying in a sac of skin, what is called scrotum, a pair of what is called ovoid testes located or placed inside the scrotum. The scrotum is a sac of skin hanging outside the abdominal cavity. What is the reason? But in some animals, the scrotum is, just in the case of mammals, is located inside the body cavity. For example, in the case of whale, elephant. In the case of echidna, one of the what is called egg-laying mammals. So whale, elephant and echidna, the egg-laying mammal, we have the testes are located inside the abdominal cavity. But in most animals, one of the mammalian characters, the testes are placed inside the scrotum. The scrotum is hanging outside the body cavity. Now here in the case of human beings, the scrotum is located just outside the abdominal cavity, hanging from the abdominal cavity. What is the reason for that one? Normally, the spermatozoa cannot, or spermatogenesis cannot occur in a normal body temperature of around 37 degrees Celsius. It needs a temperature less than the normal body temperature of 37 degrees Celsius. So, the main reason for placing the testis in the scrotum outside the body cavity is to have a temperature 2 to 3 degrees Celsius, 2 to 3 degrees Celsius lower than the normal body temperature. So in the scrotum the body temperature is 2 to 3 degrees Celsius lower than the body temperature, because the normal body temperature cannot actually allow the differentiation of the formation of spermatozoa by spermatogenesis. That's why normally the testes are placed in the scrotum hanging outside the abdominal cavity. Hence the scrotum is considered as a thermoregulator organ. Thermoregulator organ, as it regulates the temperature for proper formation of what is called actually the sperms or spermatozoa. So that is why what is happening, it is acting as a thermoregulator to form the sperms by means of spermatogenesis. That's the role of scrotum. Okay, the testes are ovoids lying inside the scrotum. I mentioned about the function of the scrotum. Why? Or actually the reasons for the location of scrotum outside the body. Now we have to go for the structure of testis. If we are taking a structure of testis, each testis is formed of an outer covering, outer connective tissue covering, outer connective tissue covering. This is called tunica albuginea, tunica albuginea. Having an outer covering, what is called tunica albuginea, which encloses what we have the testis. The testis is divided into 200 to 250 lobules by septum. So each testis is divided into 200 to 250. That is what's called the testis lobules or testicle lobules. That is by the formation of septum. And if you are taking each lobule contains a highly coiled tubular structure. 2 to 4 highly coiled tubular structures called seminiferous tubules. So we have 200 to 250 lobules, and each lobule we have 2 to 4 seminiferous tubules. Normally the seminiferous tubules form about 80% of the testicular substance. 80% of the testicular substance concerned with the production of the sperm. So outer connective tissue covering called tunica albuginea. Then by means of septa, each testis is divided into 200 to 50 lobules, and each lobule contains 2 to 4 highly coiled seminiferous tubules, the unit of testis. And nearly 80% of the what is called testicular substance is formed of seminiferous tubules. Now the seminiferous tubules are also called testicular tubules. The unit of testis the place of formation of what is called the sperms. The site of production of sperms is nothing but the seminiferous tubules. That's about the testis. If you are taking each testis is lined with what we have stratified epithelium. If you are making a cross section of one, that is what is called seminiferous tubule, it is lined with what is called germinal cells in the form of stratified epithelium, that to cuboidal epithelium. Now I'll show that one with a simple picture. What are the cells present in seminiferous tubule, the unit of testis? Now it is being formed of stratified cuboidal epithelium. If you are taking the wall, it is called germinal epithelium, or being formed of stratified cuboidal epithelium, arranged in many layers of cuboidal epithelium. Now it is being formed of two types of cells. One, Sertoli cells or nurse cells, which is diploid. Another one, spermatogonic cells, which is also diploid. Now what is the role of Sertoli cells? What's the nature? Now the Sertoli cells are elongated cells and all joined together to form a pyramid-like structure. Now I represent elongated cells which are in the form of a cone-like structure, to which a number of sperms are stuck to the surface. Why? Now they are called nurse cells. Their main function is providing nourishment to the maturing sperm, to the sperm or spermatozoa till maturation, providing nourishment. That's why the sperms, you see that on here mentioned the black colored one, and here to the surface of Sertoli cells because they provide nourishment. This is one of the functions, hence called nurse cells, looking after till maturation. Another function, secretion of a hormone, inhibin hormone. Now, this hormone actually is involved in negative feedback control of spermatogenesis. Negative feedback control of spermatogenesis or sperm production. That is, normally the sperm production is initiated by FSH hormone, follicular stimulating hormone from the pituitary. Now once there is no need to produce a sperm, now this inhibin hormone suppresses the secretion of what is called FSH, suppresses the secretion of FSH, so the further production of sperm is inhibited, hence the name inhibin. That's why it's called negative feedback control. Negative feedback control of sperm production by suppressing the secretion of follicular stimulating hormone of the pituitary gland. So one function, just nourishment, another function, suppressing what we have actually the sperm production by negative feedback control, by suppressing the secretion of FSH. Then another cell I mentioned, spermatogonic cells. These cells undergo repeated meiotic divisions. We have mitotic division. First they are increasing in number by mitotic division, and each spermatogonium is called what is known as a sperm mother cell, which undergoes meiosis to form what is called the spermatozoa by passing through different stages. We'll see later. So the spermatogonic cells divide meiotically and differentiate to produce what we have, the spermatozoa. So here represent what we have, a single seminiferous tubule lined with what we have, the germinal cells, the stratified cuboidal epithelium. There are two types, one Sertoli cells, another one what is called spermatogonic cells. Spermatogonic cells produce the sperm, and the Sertoli cells provide nourishment, and also by negative feedback controls the process of sperm production. Now what are the other types of cells present, that is in the case of testis? Let's see. What are the other cells present in seminiferous tubules? Number one, Leydig cells or interstitial cells. They are called interstitial cells because they are present in between the seminiferous tubules. Now the Leydig cells are placed in a soft connective tissue surrounding the seminiferous tubules. I represent here in the black. That means it is present in between the seminiferous tubules, surrounding the seminiferous tubules, found embedded in soft what we call connective tissue. That is number one. These cells are endocrine cells because they are concerned with the secretion of a male sex hormone androgen, particularly the testosterone. So they are endocrine cells concerned with the secretion of male sex hormone androgen, such as testosterone. Okay, responsible for the development of secondary sexual characters, also for the maturation of seminiferous tubules, etc. In addition to that one, so I mentioned here there's a black color one between the seminiferous tubules, the Leydig cells or interstitial cells. Then other cells, what we have, immunocompetent cells. They are providing immunity against what we have. Any foreign antigen that enters the testis, they are called immunocompetent cells. So in addition to what we have, the Sertoli cells, what we have, there's the gonial cells, what we have, the spermatocyte cells, spermatogonic cells. Also we have Leydig cells and immunocompetent cells present in between the seminiferous tubules or surrounding the seminiferous tubules. Here is a picture I showed. See a subseminiferous tubule, look at the picture and understand what is Sertoli cell, what is actually spermatogonic cells, and also we have Leydig cells or interstitial cells. Now the immunocompetent cells are not represented. Now we have, so this is about the testis. So far we have discussed what are the components present in the testis. Now we have to go for the accessory ducts, a paragraph. The second part of the human male reproductive system consists of the various accessory ducts associated with what we have, the reproductive system. Now, the seminiferous tubules converge to form a tubular structure that is called tubulus rectus. Now, the tubulus rectus convey the spermatozoa into another structure, what's called retive testis. It's a network of tubules present on the posterior side of the testis. Now the retive testis convey the spermatozoa into a highly coiled tubular structure, epididymis, through a number of small ducts emerging out from the testis called vasa efferentia. So from the retive testis, the sperms are conveyed to the epididymis through small ducts leaving from the testis. They are called vasa efferentia. Now epididymis, you have to know, that is for 3 marks or 2 marks question. It is a single highly coiled tubule where you have the sperms are stored temporarily. So temporary storage of sperms, that is number 1, temporary storage of sperms. Now again, the spermatozoa undergo physiological maturation. Physiological maturation that helps in what we have, increased motility and fertilizing capacity. The physiological maturation normally takes place in the epididymis for actually increasing the motility of the sperm and the reproducing ability of the sperm. So that is happening in the case of epididymis, a highly coiled tubule present on the dorsal side. Now the epididymis enters into the sperm duct, what is called the vas deferens. The vas deferens receive a pair of ducts from a pair of seminal vesicles, the accessory glands. You will see in the picture, also in the notes. So it receives actually, each normally we have 2 vasa deferentia from each side. Now they join together and they receive actually a pair of ducts from the seminal vesicles. After joining the ducts of seminal vesicle, now the vas deferens is called ejaculatory duct. The ejaculated duct normally passes through the prostate gland and reaches the last part of the reproductive system, namely urethra. So the vas deferens is called ejaculated duct after joining with the ducts of seminal vesicle. Now the ejaculated duct passes through the prostate gland, a gland normally present below the urinary bladder, into urethra. Now the urethra is normally the final part, the terminal part of the reproductive system. It is responsible for conveying both the semen and what we have the urine at different times, only a common passage in the case of male but not in the case of female. In the case of male, there is a common passage for the elimination of semen, that is a reproductive material, and urine, but it is occurring at different times, not at the same time. Then it passes through the penis and opens outside by means of opening That opening is called urethral meatus. So it is not coming in what is called accessory ducts, only the opening of the urethra is called urethral meatus, which opens outside. Now I will show all these ducts, okay, in a picture. So now the seminiferous tubules converge to form tubulus rectus from each lobule. Then from the tubulus rectus, the sperms are conveyed to radiate testis. The radiate testis is nothing but a network of tubules which convey the sperms to the epididymis through vasa efferentia, many vasa efferentia, vas efferens, singular, vasa efferentia, plural form, emerging from the testis, leading to what is called actually epididymis. The epididymis is a major structure concerned with the temporal storage of the sperms, and also physiological maturation of the sperm is taking place with the increased motility and also reproducing capacity. The vas deferens after joining with the ducts of seminal vesicle to form a ejaculative duct which passes through the urethra, and the urethra actually passes through the penis, the copulative organ, and opens outside through an aperture. That aperture is called urethral meatus. I'll show the picture now, the section of testis showing all these tubules. Now let's see what are the structures present on the inner side of what is called the testis. That means testis showing inner details. Now each testis is normally covered by a fibrous connective tissue called tunica albuginea. We have two more layers also, I am not telling that one. Now the testis is divided into 200 to 250 testis tubules. And each tubule is formed of 2 to 4 seminiferous tubules. The blue one indicates the seminiferous tubules present in each lobule. That is 200 to 250 lobules are present. Each lobule we have 2 to 4 seminiferous tubules. Now, from the seminiferous tubules, there converge a tube, what's called tubulus rectus. Now the green one converge at the seminiferous tubule, terminal part, that's called tubulus rectus. The tubulus rectus normally opens into a network of radiate testis, present dorsal. And from the radiate testis, the sperms are transported to the epididymis, a highly coiled tubule, by means of what's called vasa efferentia, what I mentioned in black color. Now the epididymis is passing towards what we call the abdominal cavity in the form of vas deferens. Now we'll see the vas deferens receives, you know that one, the ducts of seminal vesicle to form a ejaculative duct. Then we have the urethra which opens outside. That we'll see later under the male reproductive system, the entire one. So these are all the components present normally in the testis, showing we have tubulus rectus, epididymis, radiate testis, vasa efferentia, vas deferens, which is proceeding up towards the abdominal cavity. Then we have the outer covering of the testis, tunica albuginea. That's about what we have the inner details of the testis. We have to go for the glands associated with the male reproductive system, okay, and finally the external genitalia, the penis, along with the diagram. I'll conclude the next class.